So thanks very much, Regan. I think that was a good start for all of us. And now we are going to move to our next speaker, Professor Hannah Holscher. And Professor Holscher is actually an associate professor of nutrition in the Department of Food Science and Human Nutrition and a member of the Division of Nutritional Sciences, the Institute of Genomic Biology, and the National Center of Supercomputing Applications at the University of Illinois, Urbana-Champaign. So far, to multidiscipline in one person, right? Her research focus are clinical interventions and computational approaches to study the interactions of nutrition, the gastrointestinal microbiome, and health. So Dr. Holscher will actually talk about the diet, health, and the microbiome. Please, Hannah.
All right, good evening. Thank you all so much for being here. I know this has been a great conference. The days are really long, and we're right at the end of one, and you had other choices of places you could be. So thank you for joining us. First, to start with some disclosures, my employment at the University of Illinois, my grant and research support, everything in bold is specific to research that I'll be presenting in this presentation today. My speaking and travel support that I've received, and I do certainly want to also acknowledge, though, that this presentation was made at my discretion and contains information that I thought was most pertinent to share with all of you without influence. So I'm going to be talking about personalized nutrition and thinking about that from three different areas. So within personalized nutrition, we'll have a definition, and then I want to do a slightly deeper dive on some of the factors that make that up. The individual factors, dietary factors, and then microbial factors. And then I'm going to get more into how we might be able to use the microbiota as predictors of physiological responses to the diet. And then in the last part, I'll be talking about some of the work that we've done actually with dietary biomarkers and the potential for using microbial predictors of food intake. So I loved how Regan kind of teed that up, right? So again, it's not meant to replace any of the others, but there is some opportunity that it can help complement some of those already in place biomarkers. So firstly, a definition of personalized nutrition. There are several of them, but just to ground us in the same area, I do like this one, and you can read it on the screen there. I'm going to grab water just in case. All right, so thinking about personalized nutrition, I am a registered dietitian, so I've been thinking about personalized nutrition since I started as an undergraduate student. And so my training has encompassed thinking about these individual factors. So working with individuals and considering them as the treatment target. My training as a nutritional scientist, obviously brings a different lens. And then, as was alluded to in my affiliations, I really just love the opportunity at Illinois. I've been able to work on lots of different interdisciplinary teams and wear a few different hats to be able to move this research area forward. So thinking about the individual factors, humans can respond differently to the diet due to their life stage, health status, physical activity, stress, dietary habits, eating patterns, genetics, microbiome, etc. So what I actually want to ask you all to do, though, is maybe just take a second and reflect on some of the individual factors that have changed for you over the last 10 years. So I'm not going to ask anyone to share, so don't be worried about that. But I want you to take a moment to reflect deeply about how your individual factors may be different in 2024 than they were in 2014. All right, so I invited you to reflect on your personal journey because many of you, like myself, may have noted several specific factors that were different from you 10 years ago. And those factors could affect your ability or your desire or your need to undertake a personalized nutrition intervention. So I've got just one lighthearted example to provide you. So 10 years ago, I didn't know that, oh, sorry, let me rephrase that. So I have the bitter taste receptor where foods like broccoli are very off-putting to me. I know broccoli is a very nutritious food. And so because I don't enjoy consuming it, I rarely will eat broccoli in any form, especially the raw form. So today's Hannah now knows that there are different culinary approaches that can be used, like air frying broccoli. If it's marinated in some olive oil and then put into an air fryer, I will consume an entire head of broccoli. And so this is just a very lighthearted example of how you can use education and kind of exposure using different cooking approaches to help an individual move past a dietary avoidance to improve nutritional consumption, right? Because we all are, the vast majority of us are not consuming adequate fruits and vegetables, and something like broccoli, which has been studied at the University of Illinois by Dr. Elizabeth Jeffrey, I'm glad she's not in this room because she would be very upset with me for saying that I rarely eat broccoli. So that was just a lighthearted example in that regard. So again, as we consider these dietary factors in the context of personalized nutrition, there are other components that we also want to consider. So food is very complex. We have to consider both the food science and the nutritional components. But then there are individual factors that will come into play as well. Someone's religion or personal preferences for why they may choose. Reagan was also talking about, you saw on the list there, fasting, for example, that a lot of individuals will partake in. So these are some things to consider in that regard. And then there are also going to be some more nuanced food factors that will interact with the host, and then also, again, thinking about dietary patterns. So when we think of foods and those dietary patterns, those will also matter, thinking in the totality of the diet. So a simple example would be if you consume carrots with oil. So consuming a fat source with the carrots will help to increase the absorption of the vitamin A. And then some individuals will also have different genetic components, different SNPs that allow them to metabolize different nutrients. So with the broccoli, for example, depending on your genetic background, you can metabolize those glucosinolates better than others. So we have to consider the complexity of food as well as the complexity of the person. So ultimately, foods contain nutrients. They'll vary in their bioavailability in that example that I just gave. Foods also contain microbes. And then if we reflect back on what Reagan was talking about, one of the other big challenges we have in the dietary arena and to be able to make these personalized nutrition recommendations is the incompleteness of these dietary databases. So I was talking about this earlier today. Fiber is one of my favorite nutrients because I study the microbiome, and fiber is a great energy source for microbes. We have very, very incomplete data on the types of fibers. On our food labels, it says insoluble or soluble fiber. That tells us nothing about the molecular configuration that is going to be, and the glycosidic bonds that are linking those fibers that allow microbes to utilize those, and then those are going to produce subsequent byproducts. So food is complicated. A few other examples, we can think about time. With resistant starch, if someone says that they ate a banana, we need to know, what did that banana look like? Was it fresh? Was it dark green? It's going to be high in resistant starch. Or was it getting very brown and you need to turn it into banana bread? With processing, that will also come into play. The almonds example is when you grind almonds into an almond butter, they're going to be utilized as an energy source that's more or less completely metabolizable or is as predicted by Atwater factors versus consuming those whole. With temperature, we, again, if someone tells us that they ate a potato, we need to know, what form it's in, its cooking form, but we also need to take that one step further. So if someone consumes potatoes or pasta that have been heated and then cooled, were they consumed cooled, or were they reheated to change that resistant starch content? Because all of this is going to come into play. And so ultimately, all of these different factors, and then fermentation is one other thing. So as we think about the microbes that can be added into foods, you can have something like sauerkraut, for example, or let's get more complicated, even kimchi. So we're using the microbes to create these fermented foods that are already present within those plants. So that means it's going to depend on the soil and these other complex factors and the amount of salt that's added in there to allow the lactic acid bacteria to accumulate. And so not only do we not have a complete database of nutrients like fibers, we also barely know what microbes are in our food system. And these microbes, certain microbes are important and beneficial for stimulating positive immune responses. And so it is important to be able to extrapolate this out, because the more we know about our food, the less noise we'll have when we try to make these diet health connections. All right, so the last factor as we think about this would be the microbiome. You can see a schematic depicted here that there are microbes throughout the gastrointestinal tract. They will vary based on the temperature and the pH, the oxygen content. In general, they'll get more dense and diverse as you move into the colon. But an important take-home here is that microbes will metabolize those non-digested substrates, and that they will also interact with host secretions, and ultimately that can generate metabolites. One of the sessions that I had the pleasure of chairing on Monday, they were talking about the host metabolome because that was the favorite topic of the night, and they're saying estimates suggest maybe at least 10% of the circulating plasma metabolites may be microbial in origin. So again, as we think about these, and also the really important factor that each of us is going to have a slightly different microbial makeup, a fingerprint in essence, that's certainly going to play a role in personalized nutrition. So these microbes and the microbial metabolites are going to have both local and systemic effects. So they have the propensity to play a role within host health. So ultimately, thinking about personalized nutrition, we really need to understand the personalized responses to the diet. And thinking about that includes characterizing all of the individual dietary and microbial factors, and then really taking advantage of some of the more advanced analytic approaches to be able to better understand these. And so then thinking back on the opening session, for example, Dr. Diana Thomas was really encouraging scientists to even make use of the free responses in their questionnaires and trying to capture more information from study participants, because there's some individual factors that we don't even know yet are going to give us the most information about being able to put people into phenotypic groups that will have the best responses to certain dietary interventions. All right, so now I want to spend some time talking about physiological responses and how the microbiota can underlie some of these. And so again, this is a really newer area of research. As Reagan was alluding to, nutrition research around the 1920s, the microbiome field is exceptionally new. We had the technologies and the databases. This area really started to explode in 2012. When the Human Microbiome Project came out, they had been using the data for a few years before that, right? We had the molecular approaches and the computational approaches so that we could sequence and start to understand the totality of the microbiota. But even today, we do not have a definition of a healthy microbiome. And so as we think about understanding how the microbiota may be used as a predictor of physiological responses, this is very much an iterative and developing area. All right, so with personalized glycemic responses, there have been 2 large proof of concept studies that looked at large inter-individual variability in postprandial glucose responses to identical meals. They also looked at triglycerides and insulin responses. For the algorithmic development, they were able to use the totality of the data to get fairly good prediction of the glycemic responses, about 0.7 to 0.77. The first is the Zevi trial, that first 2015 Cell paper, and then Berry paper with the PREDICT study is the second reference. They weren't able to accurately predict triglycerides or insulin. What was also really interesting was that using the algorithm designed by AI versus a registered dietitian, they were actually able to create a menu of good foods that resulted in a lower postprandial glycemic response. So I am by no means saying that these algorithms should replace dietitians, but think of the power that that could have by adequately educating dietitians to use these applications and to input the data that will be most relevant to work more effectively with their patients. And then the predictive factors overall would vary by outcome. The microbiome was able to predict postprandial glycemic response, around 6%, insulin around 6%, triglycerides around 7%. Genetics was also very important, around 10% for postprandial glycemic response, and quite low for insulin and triglycerides. All right, so what about the weight loss? So thinking about weight loss and the microbiota and its relevance for predicting there, what we've seen in several post-hoc analysis studies, so going back and analyzing data that have already been completed to try to better understand responders and non-responders, is that the high Prevotella to Bacteroides ratio is associated with weight loss in 3 separate interventions. So the first intervention was using the new Nordic diet versus an average Danish diet. The second was a 6-month intervention that did a caloric deficit with high and low calcium. And the third was a smaller one, just 1 month in adults with overweight and obesity, where they did a fiber arabinoxylians versus polyunsaturated fatty acids. So looking at this high Prevotella to Bacteroides ratio, the post-hoc analyses, these were relevant factors for predicting weight loss. What I found was exceptionally interesting, though, is when you actually added in additional genetic data, specifically looking at the salivary amylase gene copy numbers, when those individuals had lower salivary amylase gene copy numbers and provided the high fiber diet, they had an even greater magnitude of weight loss. And so that speaks to what's happening is you've got more undigested starch reaching the colon because of the low amylose copy numbers, right? You're getting less digestion in the mouth from salivary amylase and less digestion from pancreatic amylase that's secreted from the pancreas, and so more exposure of the fiber for microbial fermentation. Those individuals had the greatest responses to these. So the fate of the starch would also depend on the intestinal bacteria. So those individuals, their baseline gut microbiota, this Prevotella to Bacteroides ratio, predicted weight loss, and it also had an additive effect with the host genetics. All right, so some of the work that we've done in this space is using some slightly smaller cohorts and slightly different approaches. So when we were interested in this area, we were working with my colleagues. We were also looking at an opportunity to develop algorithms that can be used in other applications. So thinking about these multivariate algorithms that are able to utilize the data available in the most appropriate way. So this is some multivariate responses using random forests. And what we found looking at an avocado consumption trial that we had completed is that microbial phenotypes were predictive of health benefits of avocado consumption. So you'll note cluster 3, for example, in blue that I've highlighted, they're getting the most benefit for systolic blood pressure. And so these phenotypes, these 3 clusters, are based on just the algorithmic selection of the different microbial phenotypes. So it's more microbes in it. I think we had about 6 microbes in each phenotype, and there was some overlap between them, but it wasn't as simple as a Prevotella to Bacteroides ratio. But we are looking at 3 separate microbial clusters being able to help to predict the health benefits. So with blood pressure versus LDL cholesterol, that was actually shown with a greater benefit for those individuals that consumed this avocado intervention for 12 weeks for their LDL cholesterol concentrations. Moving on to a different complete feeding study that I had the opportunity to be involved with. This was looking at almond consumption. So individuals, a complete feeding study, the only difference between the control and the intervention was if they were provided with almonds in their diet. And there were a number of different health factors measured in that study, but I want to draw your attention to the inflammatory markers. What you'll note here is that it's cluster 2. For these individuals in this study that had this microbial phenotype, they had the greatest reduction in all of these inflammatory cytokines. And so again, thinking about these phenotypic responses and what may be underlying some of these responders and non-responders to dietary interventions. So in summary, the personalized nutrition area and using microbial predictors is that microbial phenotypes can be predictive of health outcomes. Again, this is a really new and evolving area, but we've seen benefits, or that it can be useful for blood pressure, LDL, triglycerides, and inflammation within my laboratory and collaborators. And then others have shown fairly robust data for proof of concept with glucose, and then also post hoc analyses for those studies I showed on weight loss. But again, your take home for these data is really that this is a really new evolving research area, so please continue to pay attention because it will continue to evolve really rapidly. So in the last couple minutes, I'm just going to highlight some of the work that we've been doing thinking about dietary biomarkers in my laboratory. So as we think about dietary biomarkers, the goal here is to make this subjective information more objective. So what different biomarkers can we have? We want to bring this into focus, right? Self-reported dietary data is inadequate, and so are there opportunities so that we can add objective biomarkers from different avenues to make us more confident in the information that we do have? And so building on some of the complete feeding studies and intervention studies my group has done, we had these large data sets for individuals that had participated in a 12-week avocado intervention, around 100 people for that one. A complete feeding study where the only difference was broccoli or no broccoli. There were only 18 people in that study. Another complete feeding study that was a parallel arm design that had approximately 30 people in each of the arms, either a control arm, an oats arm, or a barley arm. The almond intervention was 18 individuals consuming almonds versus no almonds. And the final complete feeding study was walnuts versus no walnuts. So to point out here, again, I'm aggregating data from randomized controlled trials. Walnuts, almonds, oats, and barley, and broccoli were complete feeding studies done at the USDA Beltsville facility in near DC. They were monitoring these participants very closely, feel very confident in what they were consuming. So with these data, what we were interested in is using bacteria as biomarkers for predicting food intake. And so my graduate student at the time, Leila Shen, one of our computer science undergrad students, Aditya Manashara, and my colleague Roqing Zhu and I set out to see, could bacteria be used to predict food intake? And so we started with some feature selection. Marginal screening is a good way to take these complex data sets and distill them down. And then we would do classification using random forest. And that would allow us to select the most important features to move forward into the algorithm. And then we were very, very careful in our validation. This is proof of concept work. It's very ivory tower in the regard of academia versus translation, right? It was a thought process. So we did want to make sure, one, that we weren't just predicting if someone was in a study. We wanted to be absolutely certain that we were predicting when they consumed the food versus the control, and not just their participation in a oat study or being in Beltsville, Maryland. So there was a lot of validation. There was also a lot of data projection to remove the background diet, because again, remember, for a lot of these foods, the only difference was between the background diet or the control was the addition of walnuts or almonds or broccoli. So we did a lot of validations to ensure we were predicting the food and not the study. So when we published this in JN in 2020, we did in fact show that we could predict food consumed with varying levels of accuracy for these foods. So I'm summarizing some very complex data using just some percentages on the slide here. The confusion matrix are in that paper, as well as the microbes that were most relevant for predicting these foods. But to give you a high level, what you're seeing is that except for broccoli, we had really good predictive accuracy using 16S data for these. One of our reasons for we suspect broccoli wasn't as good was because of the low sample size. When we used a similar approach, it did need quite a few modifications because the data structures for metabolites are different than data structures for when you're using 16S data, and our sample sizes were a bit smaller for this one. But again, we were able to predict when the participants consumed either walnuts or almonds using fecal metabolites for this. So I should clarify, these are using the stool samples as biomarkers. So we could predict walnuts with 82% accuracy and almond consumption with 89% accuracy. The other studies, we couldn't get above the chance, and so those were not robust for the fecal metabolites. The last one was using fecal metagenomes. And so looking at the full functional and genetic capacity of the microbes, so both their functional pathways, as well as the microbes in greater detail. For this one, these same foods, we had the greatest predictive accuracy for walnuts, almonds, and broccoli. So broccoli was actually able to rise up and be relevant and be able to be predicted versus its control with the metagenomic data, where we did not have a signal for 16S or metabolites. That one of the key reasons behind this one, though, is likely, again, these are complete feeding studies. The only difference between the control and the food condition was the provision of that food, and so we could subtract those background diets and allow us to get enough of that noise removed so that the model could learn adequate information. So for the biomarkers that we've been doing, we have been able to show that the fecal microbiota, metabolome, and metagenome can be used to predict food intake, including walnuts, almonds, broccoli, whole grains, and avocados. I'm not the only person that's interested in what is the opportunity for fecal samples to be used as biomarkers. You can use, Lawrence David has been doing some really interesting work with metabarcoding, where he's using specific primers to look for the plant genes within the fecal matter. And then Sean Gibbons has been working on a metagenomic estimation of dietary intake as well that uses the metagenomic data. So they've got a preprint out on that, and we've also been looking at some of our avocado data actually using his approach, and it was quite good at predicting the avocado data. So we're excited to hopefully move some of that forward too. But these are some of the areas for the fecal biomarkers. So just in the last minute here to summarize, personalized nutrition, we need to understand personalized responses to the diet. The goal here, characterize individual dietary and microbial factors. Using advanced analytic approaches can be really helpful. And we certainly need more research on host microbe interactions. So I hope what you've taken away from this presentation is that there is a lot of interest in personalized nutrition. It's not necessarily a new area. Dietitians have been doing this for a while. We have evidence-based guidelines. That's why they can bill for treatment for diabetes, for example. But moving into more of the mainstream of nutritional sciences is a really exciting opportunity for us to make that more robust. The gut microbiota likely contributes to phenotypic responses, but we need a lot more research in this area. And then finally, we do need more biomarkers to understand diet microbiome health connections. So thank you so much for your time and attention. And if we've got time, I may be able to do a quick question here before the panel.
Yes.
Thank you.
Thanks very much, Hannah. Any questions now, or you want to take that for the panel? Doesn't seem so. Cool. So thanks very much, Hannah. Yes.